Okay, thank you very much. Thank you to the organizers for inviting me here to this nice conference. As you can see, my topic is about nanowires and specifically metal oxide nanowires. That's why I was asking questions about that. Used in a thermal gradient. This because metal oxide in general and recently metal oxide nanostructures, nanowires usually, are used as chemoresistors, gas sensors based on their change in resistance. This is usually a very simple sensor that is obviously not so performing. And our idea, our novel approach is to put them in a thermal gradient to have to make them smart, meaning basically to overcome their lack of selectivity, make a sort of electronic nose with them. Now, I would like to start explaining why we need such a device. And I will do it with two examples. There are many, but these are two examples that I think highlight why it is important in our society, in our modern society, to have such a small but, let's say, smart or powerful device. The first example is the quality assessment of fresh food that up to now is made of panels of human experts, and sometimes they are helped by expensive big instrumentation in the labs. Obviously, you can see both these approaches have the same problem. They are very expensive, not only in terms of money, but also in terms of time. And that is even worse because along the production and the distribution chain, we don't have time to waste because, fresh meat, fresh fish or vegetables don't have time to waste in assess their quality because it degrades very quickly. Therefore, we would need small devices that can be placed along in a capillary way along the production and distribution chain, and obviously they must be also cheap to be capillary distributed. Another different situation, but that for different requirements needs the same, let's say, solution is the healthcare. In this case, healthy people go to the doctor only when they feel something wrong. But because obviously you cannot go to the doctor, you cannot ask people to go to the doctor every day or every week when they are healthy. They wouldn't go, and the healthcare system is not couldn't afford that. But sometimes, or quite often, that is, I don't want to use the expression too late, but for sure, if you could monitor some parameters before, you could uh, see the first warnings of something changing much before the person itself, herself, feel something wrong. That means that we would need a screening, an early screening to send the people that have a warning from the instrument to go to the doctor that will send them to specialistic doctor to do specialistic exams, because all of these are for sure have a better performance, but again they are expensive both in terms of money and time. Therefore, again, we need something that can be capillarily everywhere in the life, in the everyday life of people, and that can do a pre-screening. Again, the solution could be a small device, in this case, again, a gas sensors, because we are working with gas sensors, that can monitor some parameters and tell us, okay, I see something wrong, go to your doctor. Maybe it's nothing, but, you know, obviously, as I was saying, on one side, we have big instrumentation in the lab that are, yes, big, cumbersome, heavy, expensive, but obviously they also have very impressive performance. They are so sensitive that they can track down, I think, less than parts per trillion. They can recognize each molecule they are detecting, and they are doing it at the same time for more than one molecule, to, let's say, for all the molecules that they are detecting. On the other side, we have devices that are really the opposite, because they have their own benefits. Let's say they are already tiny, cheap, and so they are portable and integrable even in small devices like smartphones and smartwatch. But obviously their performance is completely different. In some ways, their performance are enough, because you don't need for many applications you don't need a limit of detection, like for example parts per trillion, and they have proven to have a limit of detection of parts per billion, so it's good enough. The real big problem of resistive sensors is their lack of selectivity, because they are so good, if you want, that they detect almost every gas, almost every volatile. The problem is that obviously this means also that you don't know what they are detecting, and that is a big defect. Therefore, in general, they are used in arrays. You use an array of different sensors, and combining their responses together, you have an electronic nose. This is selective, obviously not as gas chromatography or mass spectrometry, but from a big instrument and expensive instrument, you get something that is as small as a small suitcase and is some €1,000 or dollars instead of $100,000. Our innovative approach started with this work in which we were working on normal chemoresistor, normal, let's say, metal oxide nanostructures used as chemoresistors, as gas sensors. I want to show you a little bit of the material science in this part, just because it proves that this approach can be used even with the not worth, but the not optimized materials. In this case, I was working with this material that after hydrothermal growth and a thermal annealing was getting these polycrystalline nickel oxide nanowires. And you can see that they are quite homogeneous and made after the thermal annealing of these small particles that are 15, 20 nanometers in diameter. And the sensor itself was also that not optimized. You can see here that the sensor was already very small, because when I say the sensor, I mean that small part. These are 3 sensors, and all of the chip is just to, let's say, contact out the signal. So the sensor itself is really small, and it could be much smaller. So it is really possible and easy to integrate it in any chip. And what is important in this first study is that also the response is very low. You can see here the percentage response was going a bit more than 100, over 100. So it means that the signal changed of a factor 2, not more. You can see that obviously the response was increasing with concentration. That's obvious. But what gave us the idea is this behavior. You can see that the response of the hydrogen to the hydrogen gas was decreasing with the working temperature. Instead, the ethanol response was increasing. In this case, we were working on fuel cells, so we were looking at hydrogen because of that, using steam reforming from bioethanol. So we wanted to detect both these gases. This different trend brought here there are different concentrations. So here you can see it with the same concentrations, brought us to think, okay, in general, if you look at the scientific literature, you find graphs, plots like these, and this is usually described as a sensor selective for hydrogen. This means that at low temperature, we were having a sensor selective to hydrogen, but when we were increasing the working temperature, we were having a sensor selective to ethanol. Let's say a dual selective, as you can see here, sensor. This started to make me think, because as Professor Othani said, sometimes we have concepts that are, let's say, taken for granted, even in science, when they shouldn't. In this case, I started to think to selectivity, because these three papers are three very good papers on three good journals from three good groups. And as you can see, they call in their title their sensor highly selective sensor, all of them. Now, you probably know that in gas sensors, selectivity is defined as the response to the target gas, that is usually obviously the highest response, uh, over the second highest response, that is, let's say, the most problematic interfering gas. This means that the selectivity in this case is a bit more than 2. In this case, if you look at the green bars, it is less than 2. And in this case, it's the best one, it is more than 5, 5.5. Now, I'm not questioning the fact that the numbers are small. I am questioning the fact that they are numbers. I mean, one pure number cannot give in selectivity. I think that's quite reasonable. Indeed, the only selectivity that such a device, a chemoresistor, has is this one, basically distinguishing with a resistance that increases or decreases, distinguishing the two big families of oxidizing or reducing gases. Among these big families, they are not able to distinguish anything, so they have no selectivity at all. Indeed, if we take this as an example, and if I tell you, okay, now we are sure that we are measuring one of these four gases, and here is an unknown new measurement. Can you tell me which one of these four gases it is? It's impossible, because it could be any of them with a different concentration. That is what started our approach. As I was saying before, an electronic nose basically is an array of sensors. Why this? Because, and I connect it with the problem of the monodimensional response and the definition of selectivity. If you take a sensor that has a multidimensional output, for example, an optical sensor, you have a big spectrum. That means basically that you have many channels. Here you can consider it as many different sensors. Some are selective for methane in this case, some for carbon monoxide, some are not selective, because here in this part of the spectrum you can see both water and ammonia, but basically it works as an array. Indeed, what the first electronic noses did was copying this idea, or the idea of the mammalian nose, that is also in that case an array of different sensors. You can see also in that case that different neurons, different receptors are not selective. In this case, the first neuron detects all the three volatiles, the second only one, and the other two. But combining wisely the responses from all the sensors, you can get selectivity. Now, that is the idea. The problem, as I was saying, is that the traditional electronic nose is still, even if it is much smaller than a big instrumentation, it's still too big, because nobody would buy a suitcase of 3, 4, 8 kilograms and thousands euros to do just some breath analysis. We must make it tiny inside the smartphone. That is the only way. So our idea was this: not using different materials, but using one material at different working temperature. That is why we are using a thermal gradient. Basically, using the different responses that we calculate from the sensors at different temperatures, we can obtain these. I call them thermal fingerprints. Obviously, they are really a very rough approximation of fingerprints, but you can see here that they are different for different gases. Now, this is just a way to see them. the best way to consider them, even if it is impossible to visualize them, is as vectors. In this case, they would be vectors of of length 5. That means points in the 5-dimensional space. 5-dimensional because here we chose 5 sensors, otherwise n-dimensional with a generic n. Another way to see, to visualize this is, for example, without any algorithm, encoding 3 different responses at 3 different working temperatures. In this time it was 200, 250 and 300 degrees, and normalize them and use them as the channel of a color, red, green and blue channels. In this case, you can see that we were able to discriminate the 8 different gases in a visual way, with one normalization on all the data set in this case, but since you can recognize the different colors in here, you can also apply different normalization for each gas, and in this case you can enhance the difference and help to have also a let's say qualitative quantification, because if you have calibrated your system like this, and then I give you 1 and 2 other new measurements, in this case, like before I was giving you a measurement that was a bar in that bar plot, and you were not able to tell me which gas it was. Here, for sure, you recognize which gas it is, and also this. And also you can recognize even better, once you know which gas it is, you can enhance the contrast here. You can also more or less understand which range of concentration, parts per million, it is. You are sure that it will be more than 100 parts per million and probably less than 500. It's just an estimate, it's a rough estimate, but it is obtained with no algorithm, nothing. It only needs human eyes and human brain. It's very similar to the litmus paper for the pH in liquids, but this is for gases. You can see that doing again the same measurements, they follow quite well the previous calibration. These are new measurements. Obviously, that was just, let's say, a game to see if it works in a colorful way. We can visualize these also in this way as previously, but obviously you can understand that since there is noise on these measurements, it's not easy for us to distinguish, for example, these from these to these. Now, we also have colors, different labels, but if we look at them, they are very similar. Luckily, the system that we are going to use, that is any machine learning algorithm, is able to work in all the 5 dimensions, and it is much more, let's say, accurate than our site. Here is, for example, since I cannot show you the 5 dimensions, obviously, I show you here not really a 3-dimensional graph, but let's say a section of the 3-dimensional space. In this case, obtained with a principal component analysis. That is basically just a reduction of the 5-dimensional space, just in order to show you the relationship that the points pertaining to different gases have in the space and how we can use them. Here you see that the points of different gases are quite well separated. And also, you notice another thing. They are usually laying on lines. Some are longer and straight lines, some are shorter, but they usually have this strange line behavior. This is both bad and good. It is bad because it means that we cannot use unsupervised methods. Basically, we cannot say, okay, system, do it yourself, tell me without any calibration, any training, which are different groups, because you can see they rely on distance. This means that for any unsupervised system, this is one group alone, because it is so far from the other that it would be considered one group. Here, many points of different gases would be considered one group, so we cannot use unsupervised methods. But it will be good because it will allow us also to reach a further step, that is quantification, as you will see soon. The first step is anyway the classification. That is basically after we give the system some points in order to train it, it must be able to classify, to identify, let's say, any new measurement like the previous unknown bar, and it is able to classify perfectly because all the new measurements are classified perfectly with no error with different methods. Now here we were at the beginning, we were playing with different algorithms, and here you can see, for example, one of the simplest, even if this is not the one we chose, we are working right now, but this is just to make you understand how it works. For example, in this case, it's very simple. You start with all the 8 gases and you try to, or you, the system tries to find a rule to split the samples, checking the response at 300 degrees, and it found that responses lower than 1.17 were having only these 4 gases. Instead, for responses higher, these 4 gases. And with 3 different and subsequent rules, it was able to classify, recognize all the gases. And this is just classification, understanding which gas it is. Obviously, that is not enough. And using support vector machine, that is another type of algorithm, we were able to get both classification and also quantification. This is a graph that resumes both these 2 steps, because we have classification. Basically, you see that all the points are colored matching their quadrant. That means that they are identified as the right gas. And then, since in y-x you have the actual concentration of the gas, and in x you have the estimated concentration of the gas, obviously on the diagonals we have the perfect estimation of the concentration. As you can see, the points are quite close to the diagonals. Indeed, they have a root mean square error that is very low, around, let's say, 15% for all the gases together. This means that using the same sensor but at different working temperature, from a sensor that was not able even to tell us which gas it is, now we have a system that is able to tell us for sure which gas it is and also give us its concentration with a small error. Now, all that up to here was done just by me in a lab, in I could say in a proof of concept situation, because I was measuring at one temperature, then thermalizing the sample holder at another temperature. So it was taking hours to do the measurements, and that is not what could be used in real applications. Here, to show you how it works with more technologically advanced materials. Here there is a collaboration with NIO University, the group of Professor Samsu Kim. And in this case, we were using tin oxide nanowires grown on chip directly from the electrodes. You can see here from a side or here on top. There is the space between one electrode and another space and another electron. So they are, let's say, growing from the electrodes and bridging. And also they are decorated, their surface is decorated with platinum in order to exploit the catalytic effect of platinum to improve the performance of the sensor. Indeed, in this case, instead of a response that goes up from 1 to up to 2 point something, in this case you can see that we have response values as high as 50, 55, that is much higher. In this case, you can also see how the thermal fingerprints behave with increasing or decreasing concentration. And you can see that they shift, but they keep quite well the shape. That is basically what makes work the algorithm in the five-dimensional space. In this case, we can also see another thing. When I was saying at the beginning that the problem of a chemoresistor is that it is just one number, combining together five numbers doesn't mean only to have five numbers. It means also that you have all the correlations. Indeed, in this correlation matrix, you see that the correlation bring information. Indeed, in this case, you can see the whole correlation, and you see that ethanol and hydrogen are well discriminated, separated from the other gases, and the other gases that are in a small cluster are zoomed here, and you can see that most of the correlations can can identify the different gases. Indeed, looking at the principal component analysis again, we can see that also in this case, the lines, or in some cases clouds, are again quite well separated. In this case, using sensors that are a much better performance, we were able to obtain also smaller errors for some gases. You can see we went down to ethanol to 5%, and for benzene lower than 4% error on the gas concentration. But in some cases, like toluene, probably because of this dispersion, this cloud instead of a line, is much harder to get a better error. Up to here, also, we were still working in a laboratory in conditions that are not similar to those that would be operational in real applications. If we want to go with a real prototype, we must do this very quick. We cannot have a sample holder that needs to thermalize. Indeed, we did this with two ways. The thermal gradient can be in time, temporal, or in space. In this case, I show you the first approach, that is basically using only one sensor. In this case, we really have one sensor, but we grow it in a very technological way. Basically, we have a multi-layered electrode and another one here. This thin yellow layer is a gold catalyst. In this way, we are able to grow the nanowires only from this layer. So not on top of the electrodes, but only on the side. And here you can see the optical microscope of the electrodes before and after growing the nanowires. So they are growing only like lettuce out of a sandwich. And here you can see a zoom of the nanowires between the two electrodes. And also in this case, they were tin oxide nanowires decorated on their surface with platinum nanoparticles. In this case, I must highlight that it was a collaboration with the ITIMS Institute of the Hanoi University of Science and Technology and Uppsala University. Doing this complex architecture, we were able, flowing the current, to heat the sensor by Joule effect. And you can see that since the resistance bottleneck is the matter of the nanowires, basically that is where we are increasing the temperature, because that is the most resistant point. Since the thermal inertia of these nanowires is really small, the temperature increases very quickly and stabilizes very quickly. This means that in half a second we can have all the five temperatures, or all the five responses at the five different temperatures in half a second. With this method, that is for the first time practical, we could use it because it's really quick and it doesn't need anything of the lab. We were able again to have a perfect classification, 100% of right classification and a good quantification. Again, the average error is around 15% for all the gases. The last, and in my opinion the best way, is to use not a temporal thermal gradient but a spatial thermal gradient. Basically, you see here a heater and in this case only four sensors placed at different distances from the heater. That means at different, obviously, lower and lower temperatures. Here you see the same structure with the heater and 1, 2, 3, 4 sensors once the nanowires are grown. And here you can appreciate why this system can be 0.2 of a millimeter, because it uses only one material. If we wanted to put different materials, it's really, really difficult. Also, with this system, the classification was perfect. There were no misclassifications, and in this case the mean absolute percentage error was very small. For different gases, for example, for hydrogen was less than 9%, for ammonia less than 10%. So basically, the idea is using the same material but in a thermal gradient that can be declined as temporal gradient or spatial gradient. We were able to make one let's say one material, one metal oxide, one chemoresistor, one gas sensor based on metal oxide nanowires, having perfect classification, because in all the cases it was having 100% correct classification. It was identifying the gas perfectly, and also a very good estimate of its gas concentration. So thank you very much. If you have any question, I will be glad to answer them.